everyone. I'm Krithika Ramachandran. Here at CHINTA, I work as a clinical research coordinator and a research scientist. And today we'll be talking about the clinical research that we are currently doing and we plan to do at CHINTA. Okay, to just give you a flavor of that side of things. So to begin with, you guys have already seen this slide in the last lecture, right? So giving you a continuity here. So at CHINTA, again, the focus is not on any particular disease. We are trying to target neurological disorders across all the lifespan, right? So starting from the neurodevelopmental side, where we have things like autism spectrum disorders, going on to your adolescence, adult lifehood, where a lot of the psychiatric problems come up, anxiety, depression, stress, and then all the way to neurodegeneration towards the end of the lifespan. So we are trying to target all of this, right? And the reason we want to do this, and the reason why this is becoming a problem is because of the rising trends in all neurological disorders. So let's start with looking at the neurodevelopmental side, right? So this is a study that came out in 2025, so very recent, and it uses the data from this global burden of diseases study, and they show that there has been a steady increase in the prevalence of this is particularly the autism spectrum disorder cases from 1990 to 2020. And some of the factors that contribute to this increase are, of course, even the improvements in the diagnostic practices, increased awareness of people, so you're able to recognize what actually autism looks like, environmental and societal factors, and genetic and epigenetic. So environmental factors, increasing age of the parents. Right now, people get married later, have kids later, so those kind of things. Also, genetic environmental factors like what we eat, the quality of things that we eat is going bad. So all of these factors combined are contributing to an increase in the prevalence of neurodevelopmental disorders. So kids are born with a lot more problems, intellectual disability, later learning abilities, like delayed learning, all of that problems are increasing. Now going on to the next part of it, the psychiatric disorders. Again, there is a tremendous increase in psychiatric disorders. Specifically, I'm just showing anxiety and depression here. This was also from a study that used the data from this global burden of diseases, 2019. This came out in 2022. And again, you see that compared to 1990, prevalence in 2019 for anxiety and depression have both shot up, right? And just want to highlight a few things in this very complicated graph. The one thing that people saw, this paper saw, was that there is a higher prevalence for both anxiety and depression in females. So there is a sex difference component there. And just if you look across lifespan, this particular thing, which is depression, anxiety, is in this red and orange, right? So you see that sort of starts off in your adolescent age group itself. So it starts off in adolescence, and then throughout lifespan, there's like more of this psychiatric disorder. Sure. So this is again just this is talking about the prevalence of a lot of neurological disorders. Okay, so every color is a different disorder almost. So the red and orange are talking about depressive and anxiety disorder. Then you have your schizophrenia, other mental disorders come in the yellows. Green goes to autism spectrum and bipolar disorder. Then you have your developmental intellectual disability, eating disorders, ADHD, attention deficit hyperactivity disorder. All of that comes in your blue. So every color is representing a different group of diseases, right? And your y-axis is the different age, lifespan, right? So at 0 to 4 years, 5 to 9 years, so like that, it's divided into different time point of life. And then this scale here, at 0, both left and right are talking about your prevalence. That's the number that's giving you prevalence. The measure that they use is the disability adjusted life years. It's a thing, I'm not going to go into that right now, but that's another measure of how you look at prevalence, basically. So the higher the number, the bigger the problem, the more that disease exists, right? And the reason they have both left and right the same thing is because it's divided by gender. So left is males, right is females, right? So that's your orientation to the graph. Now, when you look, I'll take the example. When you look at just 0 to 4 years, the only thing you see here is the green and blue, right? That also specifically, right? So you see the autism spectrum, that comes up, the green is there, and the blue is the conduct disorder. So how you behave, right? How you conduct yourself. So that is high here. right? That almost does not exist all the way at the 1994 years, right? Because people with autism will not live that long, right? So you don't see that there at all. So that's the first one and the last one. You can compare like that. So the point I was trying to make here now is that starting at your adolescence, you see that the psychiatric disorders, the anxiety, depression, those disorders really make the bulk of the problem. So you see a big increase in all the red and orange here, and specifically highlighting one more point, that it is more in females. So if you just look at the length of this here compared to here, it's more on the right hand side. Does that make sense now? So a lot of things. Yes, of course. And it's a lot of colors and a lot of parameters that there. Yeah, exactly. It is a complicated graph. There's a lot that you can infer from that, right? So this one is about the psychiatric disorders. And then next, going on to the end of the life, so the neurodegenerative component, right? So just taking examples of Parkinson's and Alzheimer's here, again, the prevalence has definitely increased. Prevalence is almost doubled, more than doubled for Parkinson's from 1999 to 2019 again. And for Alzheimer's also, it surged 113%. So again, more than a doubling, right? But beyond that, not just that there is an increase, there's also a prevalence for an earlier onset. So people are getting these neurodegenerative disorders earlier in their life. So that's what this graph is showing. So at 50 to 60 itself, people are now starting. So this is the 2019 time point. And at 2019, you see that people are starting to, both males and females are getting these neurodegenerative disorders at an earlier time in life, as compared to when they would get it when way back in 1990s, right? So not just more, it's also earlier. Yes. So this is actually only measuring prevalence. It's not onset at all, right? Yeah, that won't affect the onset though. It will affect the total prevalence. Yeah. Yeah, no, this is not onset. This is prevalence. So the prevalence will be more, exactly. So there will be more people with Parkinson's at 80 and 85 who are living now as compared to before, because they would just succumb to the disease beforehand in 1990s. So that is definitely a good point. So that also adds to the overall increase in numbers, for sure, right? Okay. All right. Okay. So now just another point I want to make here, right, is that this problem is actually quite close to home also. The reason being, so we have overall seen that the number of communicable infectious diseases have actually gone down over, again, this is a generic span of 1990s to 2019, right? You see that the number of communicable diseases has gone down 4.2% to 1.1%. And this, you can imagine the reasons, right? Healthcare, diagnosis abilities have improved, treatment has improved, there's better infrastructure, vaccinations have improved, sanitation, even like just a basic thing that has improved, right? So all of those reasons, we have really gotten a handle on the communicable diseases. But the non-communicable diseases, which is the bulk of neurological disorders, right? All of that, the burden of that has actually doubled again. And looking specifically at India, again, this map now here shows just the non-communicable diseases in India. Overall, you see that there is, again, this is just showing for the entire India, colors are just representing the numbers. So the red means there are more prevalence of non-communicable diseases. So just highlighting here, West Bengal, Chandigarh, Odisha, this belt here has the maximum prevalence for non-communicable. Again, the reasons can be a lot. It can be just, people are living more, like longer life, so that can add to your neurodegenerative sides, increased awareness, the risk of later childbirth, so depending on, are you talking about neurodevelopment, neurodegeneration, there can be different reasons. Okay? You have to turn it on, I think, Shami. It's not on.
Yeah. So I'm just curious that I mean, in this paper, did they tell anything about why, particularly in West Bengal and in these areas, it is more?
No,
more than
they actually did not go into that. It's an observational study, right? So also this is a way of looking at prevalence. It's not actual number of prevalence, right? So this is again this DALI, disability associated life years.
No, I mean,
that's a measure.
Whatever
right?
it is, I'm just curious that why West Bengal and these
No,
three
that's,
states,
they don't
they are
actually
not
go
even
in
the Bimaru states. I mean,
So,
the other are also.
observational study, right? It's a prevalence and observational study. So
No,
they
that's
don't
why I'm
really
asking.
get into the reasoning.
No, that's why I asked, because it's a Lancet paper I can see from here.
Yeah.
So if they have talked anything about that, no,
No, they haven't.
that's a sad story.
Because this is like, imagine the amount of data that they've analyzed and then come up with like graphs like this. So no, they don't actually go into it. It's just making the observation that it is higher. And the numbers are not very, I know the red makes it look like, oh my God, it's just so much. It's actually not that bad, right? Even
no, actually bad. If you see 3,400 per 10,000 and 2,000 per 10,000.
Yeah, 2,000 is also bad.
So they should have discussed something about it.
It's not like the other places are 200 versus you have 3,400. So it's bad throughout. It's just the colors make it look like, that is another thing you should know about, like how you present it. It can exaggerate your data or make it, So if they would have used softer color gradient, you would not have felt like, oh my God, it is such a bad thing, right?
No, that's, I mean,
it is disability.
I'm just asking about them, like the paper, they did not discuss. Okay, that's fine, that's fine. Right? Then you cannot do it.
In this part of the country, yeah. Yes.
But when they were collecting the data, they may have set some domains, right? So then while choosing a domain, they may have assumed some factors like that. Like one obviously could be the age, but then since we are talking about India and different regions which are different in
come into play. That is the other thing that would, definitely
education.
Yeah.
Okay.
Because again,
I'm curious.
years of education or educational level will also affect access to care or awareness of about a disease and things like that, right?
But we are talking about non-communicable diseases and specifically neurological disorders.
Yeah, exactly. So no, this is actually not for this is neurological disorders, but even for that, awareness definitely comes into play, right? Because let's assume there are genetic disorders, right? A person who's aware that I may give this disease to my kid may not have a kid if you're aware about it, right? So there can be education will definitely play a role in even non-communicable diseases. Yeah. Right?
Fair enough. But my question is very specific to this paper who have collected the data and mapped it out. So when they were selecting the domains, like for each state, the data that they were collecting, like what other factors they took? Did they
look back at the paper for that? Because
so
also they analyze the data from this global health study, right? So it's not they did not collect the data.
Oh, okay.
So they took a data from an existing like a big paper that was done before that, 2 years before that, and then they analyze the component India specific, whatever data was there, they analyze that and then present that. So it's already a subset of something else.
Okay.
But
so then
original
parent paper. Okay.
This is a good point. Looking at that, like what they look at and how they come to this conclusion is
yeah, because this
again, the idea of this was not to highlight,
No, I completely get it. I'm just like very curious because you see that the North Belt is all blue, light blue.
Exactly.
So then this could also be like due to the weather conditions or historical.
It can be whether it can be again the genetic component of like the lineages, right? Like the kind of lineage that exists there is very different from the West or the East, right? So that can also be a component. But
Thanks.
We'll have to look into it.
Sure.
And same thing, Shami, like maybe they may not talk about it, but the original paper might have some details about, why is more or less. Absolutely, yeah. It could be. And I think actually Parita made a good point. It could even be as simple as weather, partly, because this is also a coastal thing and this is also somewhat a more humid thing because the north is less. So who knows? It could be climate, as simple as that. But I don't know beyond that. So I will tell you to refer to the paper. Right? Yeah. No. Okay, so again, the point being that neurological disorders are increasing. The only statement I want you to take home from here, nothing else. It's more in one state or the other is not the point. It's just that it's increasing tremendously, right? And the other part here is that even though there are a lot of, molecules that are tested, a lot of compounds that are tested for potential use as drugs, if you see there are about like 10,000 compounds that are tested, when it comes to preclinical stages, it already, the number reduces. And then beyond that, the sad part is when it comes to clinical trials, the number further goes down. And then actually the things that are marketed for, specifically, again, just looking at brain disorders, anything that comes out in the pipeline, you have very limited numbers, right? The reasons are, of course, the animal models cannot really reflect the complexity of what is happening in the brain. That is one part. We don't have good validated biomarkers that can test whether the drug is working or not, right? Whether it is getting translated from mice, cell lines to humans, that also doesn't exist. And then the biggest point that comes out is that there's actually a lot of variability when you do human work, right? So a person to person, even if you match everything about, even if you just take siblings and you test, there will be something different in them, right? So that's why as you try to make medicines, a lot of these factors need to be taken into account. Okay, so now coming to the focus at CHINTA. Again, maybe this is something you've already seen before, right? So the focus of CHINTA, it's a translational, the focus is on translational neuroscience, and we do want to study any particular disorder. Like I said, we're disease agnostic, we are lifespan agnostic. So anything that we take on, we want to study, look at it at all different levels, right? Starting from the behavior, up in the behavior level, going down to the systems and the networks, further into the neurons, cellular, molecular, genetic. All the different levels want to be covered. And the idea, of course, is going from the discovery side to the delivery, right? And the other part that I just want to give you a small flavor of is what all components of clinical research we ideally want to do. Not all of this has already begun at CHINTA, but this is where we want to go, right? So when you look at clinical research, the first thing is a clinical evaluation. So the basic evaluation that a doctor does, right? You start out with that. This can include your family history, just basic, the patient history, family history. It can also be your different blood tests, just the basic blood test that the doctor conducts, all of that. Then you can have cognitive assessments. So again, because we're a neuroscience institute, just testing the cognitive ability, and we will go a lot more into the cognitive assessment part. The other thing that we've really started at CHINTA is the stem cell model. So getting samples from patients and deriving, taking the iPSCs from that, so induced pluripotent stem cells from that, and then making neurons. That is another component that we're doing. Brain imaging is also a part that comes under clinical research. Then, like I said, if we take the blood or other body fluids like your CSF, urine, saliva, different body fluids, you can do biomarker research based on that. And then finally, the genetics and genomics of any particular disease. So all of these are clinical components, are part of the clinical research, but we've not started all of this in full flow at CHINTA, but that is where we want to head. Okay. And again, just to show that different clinicians have been approached. So we are actually, so CHINTA doesn't really get patients direct, right? We are a pure research center, but we do tie up with a lot of different clinicians in different hospitals, private, public, all of that, to get our patient samples. And this is not just in Calcutta specifically, we also tie up with doctors, both nationally and internationally, right? Okay. So now switching gears and going into one particular project that we are really, that started off, kickstarted CHINTA, which is this disease called spinocerebellar ataxia 12 or SCA12. So why is it unique? SCAs, there are like more than 40, 50 SCAs that exist, but SCA12 particularly is prevalent in India, specifically in the Marwadi community in India. Okay, so what is SCA? It is, like I said, it affects the cerebellum and the spino. So spinocerebellar, that's in the name itself, right? Spinocerebellar ataxia is a movement disorder, right? So that's what the name is. Looking into the genetics of that, so SCA12 occurs because of a gene mutation in a gene called PPP2R2B. So that's a protein phosphatase gene. And the 5' UTR of that gene, which is the 5' untranslated region, there is a mutation. So you have extra CAG repeats. So you know your basic DNA, CAG, cysteine, adenosine, guanine. So that CAG, there are extra repeats. Does anyone know any other disorder that is caused by a CAG repeat? A more famous one. Have you heard of Huntington's? Right? So Huntington's is also caused by a CAG repeat. Now, what can change in, and that is also a neurodegenerative disorder, right? So what changes is which gene the CAG mutation occurs in, and even in the gene, what the location of that mutation is. So your different SCAs basically will have this repeat in different parts. So it can either be an exonic mutation, so some of the CAGs have it in the exons, some of them have it in the 3' UTR. SCA12 particularly has it in the 5' UTR. Do you know what 5' UTR is? Yeah? Tell me. Sorry, I'll give you the mic before. Is there a light?
So,
Yeah.
so we know the DNA is vector. It has direction. So 5' to 3'
Yeah.
is the translational direction. So 5' UTR means the region upper to the upstream to the 5', which is not translated
Yes.
during translation.
Yes. So it is the untranslated 5' untranslated region. UTR stands for that. So what it means is that it's the part of the RNA that does not become the protein, right? So you might think that it's a waste DNA or waste RNA, but actually what that does is it plays a lot of role in regulation. So where is a protein expressed? How much is a protein expressed? In all of those parts, these UTRs, both 5' UTR, 3' UTR, all of these regions of the DNA play a role in regulation, basically. So they have more of a regulatory role. Yeah. So the other thing about SCA12 specifically is that the CAG repeat that I said, in normal individuals, you can have about 7 to 28. So it can vary. Again, I said, right, every patient, every person, every human is different. So you can have anywhere between 7 to 28 repeats of the CAG if you're normal. But then in a disease person, what happens is there is a expansion of this. So it goes up from 55 to 78. Okay. And basic thing is, yeah, you have cerebellar degeneration. This, like I said, it's a movement disorder, so it is characterized by tremors in the upper limb, in speech. There's lack of balance and coordination, gait, so ataxia, that's why, right? Like, yeah.
So can you tell us the reason for the increase in the number of repeats?
It's a mutation, it's a genetic mutation, right?
But like, what is causing it specifically?
So it's a autosomal dominant disease.
Okay.
So you get it from the
Yeah.
parents. Yeah. Sorry, that's something I should have mentioned before. It is an autosomal dominant. So either of your parents have it, you will definitely get it. Right? You take the mic again, sir.
If the UTR is not translated, then how it affects the movement of and causes the ataxia?
So it's not just that. So this I'll answer not just for SCA12, but any disease, right? So mutations, originally what people thought is only the exons matter. Exons are the regions that make up your protein, that become your protein, right? But more and more research over the last decade or more, 15 years, I would say, is now showing that all of the region that we thought is junk, so the 5' UTR, the 3' UTR, the introns, all of that is actually playing a lot more role. So again, just we can assume that if the 5' UTR number of repeat goes from 7 to 60, right? What will happen is the ribosomes, like the signals that are going to come and bind to do the translation, you may have more or less, it just changes the dynamics of the system, right? We don't know exactly what happens. That is still unknown. We don't know how that number of the increase is actually causing the disease. That part molecularly is not yet known.
It may control the PTM or the folding of the proteins.
Yeah, it may the structure of the protein, anything can destabilize. Starting from the translation of the protein can get affected. So how much or how little is made can get affected, to how is it stable or not can get affected. It might get degenerated faster. So anything can happen.
How much repeats is needed to cause the SCA?
So people have how much the doctors have characterized so far, it's 55 to 78, right? But the number it's not a fixed number. Again, this is I won't say it's a new disease, but we're getting aware about this disease now. So right now we can say that 55 to 78 causes the disease, is the disease causing length.
Okay.
Not to say that beyond 78, maybe we just haven't seen a patient like that, I guess, so far. That's what it means, right? Okay.
Thank you.
Yeah. So, like I said, so basically it is a movement disorder, so you do start getting this. The one thing that separates it from other SCAs is that the initial or presenting symptom for this SCA12 is usually your upper limb tremors. So your hand tremors is what affects it more as compared to movement, your walking and gait and things like that. You get all of that also, but it's a later thing. So presenting symptom is usually the upper limb tremor. Okay. Okay, just a couple of videos of SCA12 patients. Right, so you do see that's, and it's also not, I forget the term for it, it's not even to both sides. So you see that the right hand was having more tremors than the left. So it's not a, whatever the term is. It is asymmetric, thank you. Okay, and then this is the same, no, it's a different patient showing the gait difficulty, right? He's constantly having the tremor also, if you observe, in the hand, and you see that the balance walking is difficult. Okay, so just going beyond SCA12 here, one of the other things that we actually want to look at is using a mobile-based app to deliver a cognitive behavior therapy for anxiety in Indian adolescents. And this is in collaboration with Cornell. So what is cognitive behavior therapy? I'm going to go very quick about this and we can talk more. You can just come and find me personally and talk more about it. So it's a short-term structured psychotherapy that is used to retrain the brain. So the idea behind that is that if you change your thought, your emotions and your behavior will change, right? Just rewiring your brain and to focus on the present and not the past. So that is basically the idea of CBT, and it's a gold standard for treating anxiety. Right, and how it helps is, of course, it identifies what gives you the anxiety trigger and what are the unhelpful thoughts. It identifies those, and then it teaches tools like breathing relaxation, thought record, and gradual exposure to whatever your fear or whatever you're anxious towards. And like I said, it actively rewires the brain. So that's the basis of CBT, very well known, very well used. Now, our collaborators in Cornell, they developed this app, the CBT-based app called Maya, which teaches a comprehensive array of these CBT skills that target common anxiety features, right? So it focuses on the skills that it focuses on are education, challenging behaviors, thinking realistically, and relaxing mood. This is a picture of how that app looks, right? So education will have different videos. It'll have things like connecting the dots and things like that. Challenging, like make a list, face your problems, that kind of thing. So it's all an app-based platform. And this app has been actually tried in a pilot study in the US in 59 participants, right? So again, small numbers, but they've just tried it out. And what they found is that this app was, I think, administered for, like the participants were told to use the app for 6 weeks. And they saw that these are just different groups, and the basic, I'm not going into the paper, the basic idea was that all of them showed a decline in the anxiety score. So they were less anxious with time as they used the app 6 weeks, and then that persisted to 12 weeks. So they just did one follow-up, which was 12 weeks. So 6 weeks after they stopped using the app, and that anxiety levels were still lower. This is just different intervention groups. I'm not going into that, right? So the idea here now for us is that we want to validate that in an Indian adolescent population. So we want to use this symptom tracking and this behavioral therapy app in the undergrad population. So actually, sorry, I used adolescent, but it's not adolescent if you're an undergrad already. So early adults or young adults, we want to use that. So we want to start with, we've partnered with Ashoka University for that. And Khushi, who will be joining us, I think, in July, will spearhead this project. Okay, so this is something that is in the works and will get started. Just giving you a flavor of that. And the last one that I want to just touch upon again briefly, because this is also not yet started, is, like Nishka said, we took a lot of time to develop the entire cognitive battery that she uses now in the SCA12 patients. There are other batteries like this throughout the world that exist, one being the TORCA, which is the Toronto Cognitive Assessment Battery. So we want to see if that battery can be repurposed in Indian population to measure their cognitive health. So for that, so again, this TORCA exists. It's done in Toronto. This is one single battery instead of different tests that Nishka has to do. One battery that tests all the components that she was talking about: orientation, memory, delayed recall, recognition, visual, spatial, executive language, everything that she talked about. It can, in the population in Toronto, it was able to detect early or mild stages of dementia, and it's done in that way, right? So what we now did is tried to adapt that to an Indian population. So for example, TORCA has this component about sentence comprehension, right? So this is the question here. The girl who is kissed by the boy is blue. Can someone tell me which out of this picture represents this sentence? Right? The first one. Great. Of course, you all had no problem in figuring that out. But the content is a bit explicit for an Indian audience, right? So we are now trying to change that to exactly testing the same thing. The girl who is pulled by the boy is blue, right? We may think it's silly, but imagine we're going to use this in an adult, elderly Indian population, right? So we have to make it culturally appropriate. So that is one kind of change. Another thing, this is testing the semantic knowledge. So tell me which picture relates best with the picture in the center. Right? It's an iron board and an iron, right? So we know that association, you can all make that association. But some of the other features here, for example, that maybe not something an Indian household is or people are aware of, right? So what we did, we just simply changed it to a gas stove that we know. That's also a gas stove, right? But just a stove that we know. And we changed the association. Now the association is a pot and a stove, right? And other things like a knitting needle, people will know what that is. Again, this can be a questionable thing, right? A chair, things that Indians will understand. So right now the process that we are going through is how to adapt that existing test to an Indian population. Now that that part is sort of done, we are waiting for the approval from the Toronto people that, whatever changes we made still works. We'll validate that in first healthy people to just see whether you're able to answer the test. Same age group, right? If it's an older population we want to target, we will use that in that population. And then pilot it in a neurological disease, not necessarily SCA12, because Nishka already has done all the work with her battery in that, but maybe any other disorder. Could be stroke, could be something else, right? Then again, follow sort of the same thing that she already talked about to go into that study, right? So I'll leave you all off with, again, the core philosophy of CHINTA being, it's not just about one level or one disease. It is any disease that we pick, study that across all the levels, right? So SCA12 is our favorite example as of now. It's our golden child, right? We're looking at the SCA12 patients. Nishka is going to their houses, like getting all the information from them. We are looking at the stem cells. We're looking at trying to make zebrafish models from that. So really understanding the mechanism of the disease. A lot of the questions that you guys ask that we don't know. How many repeats causes the problem? Any of those things, we really don't know much about that disease. So that is really an opportunity that we see, that you can come in and ask any kind of question about SCA12, and CHINTA will give you the platform to address that, right? So that's sort of where I want to leave you. So when you ask the question, ask it across all biological stages. This is our clinical team. We're a short group, right? Small group right now. Khushi will join us soon. We have Nishka, who's been brilliant. Dhiman is a clinician, psychiatrist who's working with us on a lot of the TORCA and other projects. And then I'm just here to guide them and support them and not do any work. All right, thank you. Any other questions? We're already a bit over time, so you can also find me and come and ask me later.